All right, good afternoon, everyone. I think we'll make a start. So we'll probably start off with the attendance pin. So obviously this week we're continuing to look at the adaptive immune system. On the lecture on Monday, we were looking at the T-cell receptor and the B-cell receptor and how a VDJ recombination can generate that unique antigen receptor. So just want to stress the fact that obviously that VDJ recombination is what's going on during developing lymphocytes, and that's before they've ever even encountered antigen, and that is what sort of gives us that ability to recognize a whole host of antigens and potential pathogens. So we'll be building on that story today, and we'll start to look at T cells, but also the major histocompatibility complex, which is also abbreviated to MHC. But before we do that, let's do the attendance pin, which is 8249. So that's 8249. Okay, so just in terms of a little bit of revision of what we've been talking about over the last 3 weeks. So by now you'll know that dendritic cells are these professional antigen-presenting cells, and they sort of link that innate and adaptive immune system together. You'll also have a good appreciation of the clonal selection theory, as well as building on the characteristics and components that compose the adaptive immune system. And then obviously last week we looked at T-cell receptors and B-cell receptors and that generation of diversity and the structure of the B-cell receptor and the T-cell receptor as well, and also understanding how VDJ recombination, as I just said, generates that whole host of, basically that diversity in terms of the antigen receptor and how that can essentially give us memory and tailor our immune response. But also, it'll be good if you could start to describe the differences, the key differences between the B cells and the T cells, and list ways in which they're similar, but also that they're different. So there's a little table here that you can just start to populate as we go through these lectures, and just fill that out and test your knowledge on what we've been talking about over the last few lectures. So in terms of learning outcomes, what we'll look at today is T cells, but also we'll have a quick look at their co-receptors, so the CD3, CD4, and CD8. You'll also be able to understand the major histocompatibility complex. Now, this is also known as the human leukocyte antigen, or HLA, so you can often hear it referred to as both. You'll also gain an understanding of how MHC actually facilitates that interaction between the T cell and antigen-presenting cells. And then we'll also look at the terms MHC restriction as well as superantigen, and give some examples of that. So if we look at the key differences between B cells and T cells, so this is building on what we talked about on Monday. Remember that B cells, they will detect antigens that are extracellular and also in their native conformation. So remember that obviously antibodies will be secreted and they will work in that extracellular environment, and they can bind to things like pathogens, viruses. So if you bind to a virus, you'll get an immune complex being formed which can be cleared, and also you get that whole sort of promotion of opsonization as well, and phagocytosis. On the other hand, we have T cells, so their antigens are peptide fragments. Remember, they have to be processed and then presented on an antigen-presenting cell. And the T cell will only see antigen when it's bound to an MHC molecule on the surface of an antigen-presenting cell. So that's a key thing to remember. And that the T cell antigens are peptides, and that the T cell can also recognize extracellular versus intracellular antigen. And we'll go through a couple of those pathways today. So this is just looking at the structure of the T cell receptor, so you'll remember this from Monday. So you can see that here it's comprised of an alpha chain and a beta chain. You've got your one antigen recognition site on the top. It has these three complementary determining regions that you can see here that comprise that antigen binding site. And then we also have these important co-receptors. So we've got CD3, which you can see around here. And you see that it has these long cytoplasmic tails that help in kind of transmitting that signal across the membrane, so getting that intracellular signaling going. And you'll see that it's composed of gamma, delta, epsilon, and zeta chains. And that's really sort of important in that signal transduction across the membrane. And CD3, it stands for cluster of differentiation. So you'll hear a lot of immune molecules called CD and then a number. So that's what it stands for. So as we mentioned, the T cell will recognize peptide antigens that are bound to MHC, and then these have been processed and presented on the surface of an antigen-presenting cell. So in terms of the antigen presentation, this is kind of a very sort of basic diagram of how that works, but it's the antigen processing that is kind of generating those peptides that we need to be presented, and presented on MHC. So essentially, we have our peptide, it needs to be degraded, or sorry, we have our protein, it needs to be degraded into peptides. It can then be presented on MHC to a T cell receptor. But how exactly do these fragments of bacteria get processed into peptides and then presented on MHC? So this is through this whole antigen processing and presentation pathway that we'll start to touch on today and move on over the next few lectures. Now, just taking you back to this diagram of hematopoiesis. So we've got our common lymphoid progenitor up here. So this is just looking at the aspect that deals with the adaptive immune response. And you can see that we give rise to our B cells, our T cells, and also our natural killer cells, but we'll leave these out of it for the moment for another lecture. So we can see that our B cells can differentiate into plasma cells that will secrete the antibody, and our T cells will differentiate into either CD4 or CD8 T cells. Now, CD4 and CD8 T cells, they get their name by the co-receptor that's on that T cell. And this is obviously CD4 or CD8. So CD4, which is cluster of differentiation 4, this obviously helps to identify the CD4 population, and they will recognize peptides that are presented on MHC class 2. And CD8, so the cluster of differentiation 8, which is found on CD8 cells, they will recognize peptides that are presented on MHC class 1. So we have two co-receptors, CD4 and CD8, one on CD4 cells, one on CD8, and then we have two different types of MHC. We have MHC class 1 and MHC class 2. And the levels or the ratios of CD4 and CD8 T cells in the blood, they kind of vary depending on the type of infection that you want to fight. So it can be a good indicator to sort of know these levels and see how they might fluctuate in the blood in response to diseases or infection and things like that. So if we look at the two cells, the T cells, in a little bit more detail, so CD8 and CD4, so they're determined by the co-receptor. Now, CD8 cells are often called cytotoxic T cells, and these play a really important role in recognizing and damaging, or basically destroying, eliminating infected or damaged cells. So if a cell is virally infected, the cytotoxic T cell can eliminate that. Likewise, if you have a damaged or transformed cell, so a malignant cell, the cytotoxic T cell can recognize that and eliminate it. The CD4 cells are often referred to as T helper cells, and these are quite often referred to as the master controller because they signal to other aspects of the immune system using cytokines. So remember that cytokines are all important signaling molecules, and the CD4 T helper cells help to regulate other cells. Now, when it comes to CD8 T cells, there's just one, so it's quite easy to remember. We've got our cytotoxic T cell. Now, when it comes to CD4 T helper cells, you'll see that it can further differentiate. So we have Th1, Th2, Th17, follicular helper cells, and Treg cells that all play different kind of roles in various immune responses, and we'll start to uncover those as we go on through the next set of lectures. Now, when it comes to CD4 and CD8 cells, we can have a look at what they're actually doing once they become activated. So if we look at the CD4 T cell, so let's say we've got a dendritic cell that's presented antigen to our CD4 cell, so that's activated that T helper cell. So that T helper cell can now go on and help or aid other cells. So the CD4 cell, it can, if it's presented with antigen via a macrophage, it can help to activate that macrophage. So let's say the macrophage has phagocytosed some bacteria. It can activate it, and the macrophage can now go ahead and destroy the bacteria that it's phagocytosed. We also get this release of cytokines from the CD4 helper cell, as well as the activated macrophage. So it can kind of activate that macrophage and the bactericidal activity from that macrophage. If we have a B cell that has recognized antigen on its B cell receptor, and it has then processed this antigen. The CD4 T helper cell can activate it and can cause it to differentiate into a plasma cell, which can then secrete antibody of the same specificity. We have, up here, we have our cytotoxic T cell. So obviously this is recognizing, in this instance, a virally infected cell that is presenting that viral material on its surface on MHC class 1. Now the CD4 helper cell can provide help to that cytotoxic T cell to give it permission to kill the virally infected cell. So you can see that these are the roles that they're taking. So essentially you see why the CD4 T helper cell is named as a T helper cell, because it's providing help to the other cells, and also why it's the master controller, because it is signaling and communicating with a lot of different cells. So how do dendritic cells present antigens to T cells? So this is using this major histocompatibility complex that we talked about. Now this is a very large cluster of genes, and it's highly polymorphic, and it's present in all vertebrates. As I mentioned before, it's also known as the human leukocyte antigens, or HLA. And if we think about what the function is of these genes, it's essentially to encode these MHC proteins that will present antigen to our T cells. As I mentioned, it's a very highly polymorphic gene loci. And this is actually what gives all of us our individual sort of immune system, or our own unique immunological fingerprint, or like a barcode, as it were. Also, a whole variety of diseases and traits have been associated with the MHC alleles. Examples of this are rheumatoid arthritis, diabetes, inflammatory bowel disease. And one thing that I just wanted to note is there's a particular HLA variant, which is known as HLA-B27. This plays an important role in ankylosing spondylitis, but it also confers protection against AIDS. So you can see how it's playing quite an important role in a variety of different diseases. Now, these were a set of experiments that were done sort of originally in the 1970s, but it was linking MHC essentially to scent or body odor and sexual preference. So in 1976, there were some researchers, and they'd shown that you can have a sexual selection mate choice by different MHC, and obviously if mice are mated with different MHC genes, they'll produce offspring that have a much more robust immune system that is able to target a wider variety of diseases. We also know that MHC can produce a distinct sort of body odor, and this actually helps to serve as a genetic fingerprint. And furthermore, there's some research that has shown that there's mate selection, when we actually can smell this. So what they did is they took sweaty t-shirts, and they showed that there was a preference for people who had differing MHC genes. So obviously, you want to mate with someone that has significantly different MHC to you in order to give your offspring the better chance at having a more robust immune system. So in one way, MHC are really crucial genes. They're determining not only your immune system and your health and that kind of thing, but also potentially your relationships. And so that's all playing a role there. And that's the paper, if you're interested in looking at it in more detail. So we start to look at the structure of the major histocompatibility complex, so MHC class 1 and MHC class 2. So you can see we've got MHC class 1 here. This is what is binding to the CD8 T cells. And then we have MHC class 2 binding to our CD4 T cells. So when it comes to MHC class 1, you can see we've got our antigen in red here, and it's binding in this peptide groove. We've got heavy chain. So the heavy chain in MHC class 1 is alpha 1, alpha 2, and alpha 3. And the peptide groove is between alpha 1 and alpha 2. In MHC class 2, we've got an alpha chain and a beta chain, and the peptide groove is between alpha 1 and beta 1. So very similar structure, but slightly different. An easy way to remember is MHC class 2, it has two chains, okay? MHC class 1, it's got this heavy chain, and then it has this beta 2 microglobulin. So that's an easy way to remember it. If we look at the actual structure of the human MHC, so this is the gene loci here. So you can see we've got class 1 and class 2. There is class 3, but don't worry about it. You don't need to know it in terms of these lectures, so we'll just focus on MHC class 1 and class 2. So you see for class 1, we've got HLA-A, B, and C, and then we've got a little bit more complicated structure for class 2. But they're highly polymorphic. So what this means is that within a population, we have a whole variety of different genes or alleles within that population. And that's shown in these graphs here. So this is data from the WHO, and it's just showing the number of different HLA proteins that they categorized. So really, the kind of take-home message is that if we were to check all the MHC for everyone in this room, everyone would be different. Everyone would be unique, unless you were very closely related. And you can start to link that into things about transplantation and things like that, and graft rejection as well, because it's all linked to this major histocompatibility complex. So looking at it a little bit more detail, we've already mentioned that it's highly polymorphic within a population, so that's kind of what's summarized at this diagram. But then we also have this polygenic aspect as well. So within an individual, we'll have a different set of related genes. So when you start to combine this polymorphism and this polygyny as well, then it really gives us a great potential to produce a very diverse set of MHC molecules within an individual, but also within the greater population as a whole. Now, when it comes to the lab, obviously if you do work with mice and like mouse models, you want to sort of be able to control that. So what they've done is they've done a lot of inbreeding with mice strains to make sure that we have MHC genotypes, so we can kind of remove that variability, so we can kind of get identical MHC on certain mouse lines. MHC alleles are expressed in what we call this codominant fashion. So what this means is the alleles that we'll get from our parents are expressed equally, and this helps to maintain diversity through that population. So that's just an important thing to remember as well. So we've already said about how MHC is unique to each one of us, but how did we kind of come to understand that? And that was actually done by this man here, so Peter Medawar, and he was working in the hospitals in the 1940s in Glasgow, and they were treating pilots from World War II that had had serious burns. So they were performing skin grafts, and obviously they were seeing rejection. And he came out with this paper here with one of his colleagues, The Fate of Skin Grafts, Skin Homographs in Man. And really what they were showing is that there's this diversity in MHC. So this was obviously having an effect in transplantation. But ultimately, MHC is providing you with that diversity, that flexibility to respond to a whole host of infections, for instance, pandemics that we've not even encountered yet. But it also plays an important role in graft rejection, because that's what is determining self from non-self. So why do we need this MHC diversity? Well, I've kind of already said that. It's because it's the MHC molecules which kind of enable this immune surveillance. So therefore, the more variability we have within our major histocompatibility complex, the greater the diversity of antigen presentation that we can achieve. Now, there's always a risk of a new pathogen emerging, so obviously you only need to think back to COVID-19 that we're still sort of on the tail end of. But ultimately, if all of us responded the same way to a pathogen, you can imagine that's going to have pretty disastrous and dire consequences for us as a species, because if a new pathogen comes along, it could potentially wipe us all out because we all respond the same way. But I can guarantee if we released a virus into this room now and we all got infected, some of us would get probably pretty sick, some of us might just get a mild cold, others would probably not get anything at all. So again, it's time back to that thing we mentioned in the first lecture, that we all know that friend that probably never seems to get sick. And that's really down to our MHC genes. Now, it's this MHC polymorphism that affects the antigen recognition. And essentially what this is doing is it's influencing the peptide binding and the contacts between the T cell and the MHC molecule. And it's this variation in the MHC molecules that really kind of occurs within the peptide binding region. So we're looking at a top view of MHC class 1 and MHC class 2 here, and in red it's showing you the variability. So you can see that the variability is all within alpha 1 and alpha 2, which is in the peptide groove, or all within beta 1 for MHC class 2, again within the peptide groove. Okay? So it's really that variability that's determining what antigens and how the antigens are being presented to T cells. Again, this is just a little diagram to summarize what we were talking about before, but essentially here you're being shown 4 different MHC haplotypes, so in the green, yellow, red, and blue. But let's say we've had a period of relative stability, and then we get some sort of pandemic or an epidemic, and basically all of the heterozygotes survive, so these homozygotes are eliminated. So we have this kind of haplotype expression within our population. If we then get another period of peace, we can kind of get an expansion in our population. But then if we were to encounter a pathogen again, so let's say here, it wipes out everybody except the blue MHC haplotype. But ultimately, what you can see is that it's the infections that are kind of having an impact on the MHC expression. But regardless of this, we're still getting all 4 haplotypes of MHC still surviving in the population despite the presence of the pandemics. But you can see how infection is having an effect on the MHC haplotype expression as well. So if we look at the key structural differences between MHC class 1 and 2, so we've already sort of mentioned we've got the alpha chain and the beta-2 microglobulin, but the peptide binding domain for MHC class 1 is between alpha 1 and alpha 2. For MHC class 2, it's between alpha 1 and beta 1. When it comes to the binding groove for MHC class 1, that binding groove is kind of closed at both ends, so therefore it will hold a smaller peptide, which is about 8 to 10 amino acids, whereas with class 2, it's open at both ends, so it kind of hangs out, a little bit like your hand would be grabbing a stick, and it can hold a peptide of about 18 amino acids. When it comes to MHC class 1, the peptide in the groove is anchored at both ends, and it kind of arcs out, so it kind of pops out a little bit. But within MHC class 2, it's held constantly at a relative sort of similar height. So that's really kind of the structural differences between the two. So in terms of MHC class 1, we've already said it binds peptides that are about 8 to 10 amino acids. And it's important to remember that during homeostasis, so when everything is well in your body, the MHC class 2 is constantly expressing peptides. So whether that's self-peptides or whatever's going on in the cell, it's constantly just showing to the T cells what every cell has in its content. So that's normally going on all the time. And then obviously I'm just stating down here that it's the polymorphisms that affect this binding of peptide within the MHC peptide group. And that's just showing you a top view as well. So when it comes to MHC class 2, as we said before, this has a slightly different conformation, and it can hold a larger peptide within that groove. And you'll find MHC class 2 on your professional antigen presenting cells. So putting them up side by side, so what I said, MHC class 2, this will be found on your professional antigen presenting cells, so your dendritic cells, your macrophages, your B cells that are professionally presenting antigen to the T cells. And then we have MHC class 1, which is expressed on all your nucleated cells. So pretty much all your cells, apart from your red blood cells, because obviously they don't have a nucleus, and also neurons as well. So if we start to look at where the antigen is being processed, so if we consider a body cell that's been infected, that antigen will be degraded within the cytosol, and it will be presented on MHC class 2. Whereas in professional antigen presenting cells, we're looking at extracellular antigen. So we've already discussed about how that will be taken up by phagocytosis, and they will then be processed in vesicles. So remember, we were talking about the phagolysosome and things like that. And then that will be expressed on MHC class 2. And we'll talk about antigen processing sort of in future lectures as well. Now, I wanted to bring in the importance of MHC restriction, seeing as we're talking about MHC. And really what this means is that when you get this TCR-MHC complex forming, the T-cell receptor isn't just recognizing the antigen, it's recognizing it in context with the MHC, so it's recognizing the MHC as well. So in this experiment, we've got a TCR, it's recognizing self-MHC as well as its cognate antigen. Everything's okay, we get recognition. So let's say we take that same T-cell receptor. We take the same antigen, but we have a different MHC. We don't get recognition anymore. And if we take the same T-cell receptor and the same MHC molecule that we started off with, but we have a different antigen, of course, we get no recognition because it's not its cognate ligand. But what I'm trying to get across, the fact is that the T-cell receptor, as well as just recognizing the antigen, recognizes the MHC as well. So think back to what we're talking about, graft rejection as well, because obviously that's playing an important role in that as well. So moving back to this picture before, in terms of antigen processing. So for antigen that's actually acquired within the cytosol, it's going to be expressed on MHC class 1 to CD8 T cells, or cytotoxic T cells. Whereas for antigen processing cells, we've got the extracellular antigen being taken up, being degraded in the vesicles, and being presented on MHC class 2 to CD4 cells. Okay? So let's have a look at a little bit more detail about this antigen presentation and sort of processing that's going on. So for MHC class 2, this is a very simplified diagram. We're getting some extracellular antigen. It's being taken up in a vesicle by phagocytosis. Ultimately, it will be degraded, and then it will be loaded onto MHC class 2, which will then move to the cell surface. And this is obviously mediated by this increasing acidification that we talked about in the vesicles. So you think about the phagosome merging with the lysosome to form the phagolysosome, and the pH drop and how that material can be degraded. So let's start to look at that in a little bit more detail and what's going on stepwise. So in terms of MHC class 2, these will be generated and synthesized in the endoplasmic reticulum, so you can see that here. And you'll also notice it's associated with this green molecule, which is known as the invariant chain. And the purpose of the invariant chain is that it stops any peptides actually binding to MHC class 2 before it reaches the site where the pathogen breakdown of the peptides are. So you'll see that it gets secreted into a vesicle, and then what happens is the invariant chain is broken down so that it leaves just a small portion, which is known as the clip fragment, actually in that peptide binding groove. And that clip fragment, again, will stop any peptides binding to MHC class 2 too early. So this vesicle with MHC class 2 will bind with a vesicle where we've endocytosed or we've phagocytosed pathogen, and it's been broken down. And then you have this partner molecule, which we'll talk about later, but HLA-DM. And really what this does is it catalyzes the release of the clip fragment from MHC class 2, and it allows the MHC class 2 to take up antigen. And then once that MHC class 2 has taken up antigen, it can be presented on the surface of the cell. So just to sort of zoom in a little bit more on the invariant chain. So you can see here we've got MHC class 2 associated with the invariant chain. Initially it's cleaved at this end, so it just leaves behind this fragment, and then we get further cleavage as well, so that we're ultimately left with the clip fragment. So the invariant chain forms a really important role in stabilizing that MHC class 2 before it's bound antigen. And then also the clip fragment prevents peptide binding too early, so that we only get peptide binding once we're in a vesicle with peptide. And it helps really deliver that MHC class 2 into these endocytic vesicles where it will be able to bind peptide. So going back to that diagram, obviously we've got an MHC class 2 invariant chain. It's going off into its vesicle. We're getting the sort of cleavage of the invariant chain so that we're just left with clip. We're then getting fusion with an endosome, so we're getting an MHC in presence with the antigen. And this is where HLA-DM comes along, and it actually is important in regulating that, essentially what is an exchange of the clip fragment for antigen. And then once it's got that, it can then go to the cell surface. But it's important to remember that all of the proteins that are involved in antigen processing and presentation tend to be encoded by the same genes within the major histocompatibility complex. So that's MHC class 2. So what's going on with MHC class 1? Well, in MHC class 2, the antigen is being broken down in that endosome, so it's been taken up by phagocytosis and it's being degraded by that increased acidification. But obviously for peptides that are in the cytosol, it's a little bit more different. So this is where we need to rely on the constitutive proteasome and the immunoproteasome. So what's happening is the constitutive proteasome is continually processing all of the self-antigens within a cell during homeostasis. So it's doing that all the time, and that's why you're presenting on MHC class 1 all the time. So it's that kind of self-check over. You're just saying to the T cells, like, am I okay? Am I okay? And they're just looking for anything that's out of the ordinary. Okay? Now, when you get an infection, we obviously get an increase in that proteolytic activity, and this generates what's known as the immunoproteasome, which you can see here. And this actually processes pathogenic antigens that may be in our cell because of a viral infection, let's say. So you can see that on the diagram here, we have our proteasome. It's processing the protein and chucking out all these little peptide fragments. So this is within the cytosol, but it needs to get to the endoplasmic reticulum where the MHC is. So we then have a need for this transporter associated with antigen processing, which is known as TAP, and that sort of helps to transport these peptides from the cytosol into the endoplasmic reticulum, where it can then bind to MHC class 1. And this is kind of a diagram of what's going on in a little bit more detail. So on the left-hand side, we have our newly synthesized MHC class 1 alpha chain. It's being stabilized by calnexin. We then have a whole variety of other chaperones. So we've got calreticulin and this ERP57, and they help to stabilize this MHC beta-2 microglobulin complex here. And then what happens is we have our proteasome, which is chucking out peptides. These are being transported into the endoplasmic reticulum by the TAP, as we just talked about. They can be further trimmed so that they can fit within that antigen or that peptide groove on MHC class 1 by this ERAAP. And what happens then is that we have a molecule called topazin, and this is binding not only TAP, but it's also facilitating in that delivery of peptide that's been processed here so that it can actually fit into the peptide binding group. And then once it's, we've got MHC class 1 associated with our antigen or our peptide, it can then go off and it can move through the Golgi apparatus until it's eventually released onto the cell surface. Now this diagram is just kind of summarizing that in terms of antigen processing and presentation. So we've got MHC class 1 and MHC class 2. So MHC class 2, we're talking about extracellular antigen that's been taken up by phagocytosis. It's in our endocytic vesicle. It's been broken down into small peptide fragments by this increased acidification. We're getting our MHC class 2 that's being made in the endoplasmic reticulum. It's being secreted in a vesicle. This will then bind with the endosome, and without going into the detail that we saw about before, essentially we'll get antigen being loaded onto MHC class 2 and it will be presented on the surface of that antigen presenting cell. We then have MHC class 1. Bear in mind this is going on all your body cells all the time. Let's say we have intracellular antigen. It'll be degraded by the proteasome. This will then, the peptides will be moved by the TAP into the endoplasmic reticulum, where it will associate with MHC class 1, and this will then be presented on the cell surface. So hopefully what you'll realize today is that we've got MHC class 1 and we've got MHC class 2, but essentially MHC class 1 will present to CD8 positive T cells, and MHC class 2 will present to CD4 positive T cells. So once that peptide has been presented on MHC class 2 and it's being shown on the cell surface, that's when it can be, it's available for the T cell to come and recognize it. So MHC class 1, that can be done on any cell effectively. So let's say we've got our target cell here, it's presenting that viral antigen on MHC class 1, so it's picked that up, it's saying I've got a viral infection. That'll be recognized by your CD8 T cell, so your cytotoxic T cell. which will bind to it and then obviously, hopefully, eliminate that cell. In terms of co-receptors, we have this important CD8 co-receptor. So you can see here it plays an important role in stabilizing that interaction. And also it plays an important role in transmitting the signal across the cellular membrane. So it's a kind of stability and intracellular signaling role that it plays. When it comes to MHC class 2, you can see that we've got this on an antigen-presenting cell. It's being recognized by your CD4 helper cell. Okay. So we need to talk about cross-presentation. So there's certain subsets of dendritic cells that actually perform cross-presentation. So as we've already talked about today, we have our professional antigen-presenting cells. So what these do is these internalize extracellular antigen. They present it on MHC class 2 to immature CD4 T helper cells. We also get appropriate co-stimulatory signals, which we'll talk about later, and this will activate that T helper cell. So that T helper cell can go off and do the functions that we talked about earlier on during the lecture, like activating the macrophages and providing the necessary help. Likewise, we've also talked about how MHC class 1 can present intracellular antigen from the cytosol, so whether this is a viral infection within a cell, and it can present it to mature cytotoxic T cells, which will then eliminate that infection. But that leaves us with a little bit of a dilemma in how the immune system can actually activate an immature CD8 T cell to eliminate intracellular pathogen. So let's say, that would require also our antigen-presenting cell to become infected with the same virus. But viruses tend to have a tropism. They're going to infect certain subsets of cells. So, a virus is not necessarily going to infect your antigen-presenting cells. So what happens here? How are we going to actually sort of alert the immune system and activate that immature CD8 T cell? So this is where we come across cross-presentation. So what happens here? It's shown in this diagram here. We've got our dendritic cell in yellow. It's picked up extracellular antigen. It's presented it to a CD4 T helper cell on MHC class 2. It's activated that CD4 T helper cell, but in the process, it's become licensed. So we say the dendritic cell is licensed, and by that, it kind of gives that dendritic cell permission to divert that extracellular antigen that was going to be presented on MHC class 2 to MHC class 1. So it's now presenting extracellular antigen on MHC class 1, which can be recognized by the immature CD8 T cells. They can become activated with help from the CD4 cell to become an activated CD8 cytotoxic T cell that can then go off and recognize that viral antigen that's being presented on any body cell on MHC class 1 and then destroy it. So that's just an important idea to get around your head, that we do have that. Obviously, if it's such an important role, why don't we have it on all antigen-presenting cells? Well, obviously, expressing antigen on MHC class 1 opens you up to the potential destruction by a cytotoxic T cell. So we kind of try and keep it limited. And then I wanted to talk about superantigens. So superantigens are a pathogen product, and they're there to misguide our immune response, okay? So they help to, they actually stimulate a primary T cell response, but they're produced by either a bacteria or a virus, and so we've got some examples up here. And they provoke an immune response that's actually beneficial to the pathogen, but not to the host. So you can see they kind of bind across the MHC antigen TCR complex. So they're kind of binding they're not actually within the peptide group. So it's really important to realize that superantigens, they're recognized by the T cell without being processed first into peptides, okay? And we get this stimulation of the T cell, but it's not going to mount an effective immune response because we're not getting presentation of that peptide. In effect, we're causing this massive cytokine storm. There's a huge release of cytokines. This is causing, systemic body-wide toxicity, and it's actually suppressing the adaptive immune response. So in a way, the pathogen is kind of hiding out in the chaos that's going on in the immune response. So it's kind of diverting the immune system to do something else, but actually recognize it. And an example of this is Staph aureus, where you get this toxic shock syndrome, which is actually being mediated by this superantigen that is kind of triggering this cytokine storm. So that's it for the topics that we're going to cover today, but there's a quick quiz. So if you want to go and poll everywhere, and just have a go at that. I'll give you a few minutes. All right, it looks like you're kind of beginning to get there. Just give you a few more minutes.
Is it right after we're going to do really quick on Zoom with the Life Sciences Society?
Sure.
Quick.
All right, so which one is MHC Class 1? So it looks like the majority of you got this. So remember, an easy way to remember it, MHC Class 2, it has 2 chains. So that looks perfect. In terms of MHC Class 2, it is, of course, this one. MHC Class 1 is recognized by CD8 T cells. Okay. What is the role of CD8 cytotoxic T cells? So it's a killer cell, so it's to kill damaged and infected cells, or perhaps malignant cells, transformed cells. So that all looks perfect as well. The role of CD4 cells, so it's the T helper cells, so it's to do that cytokine signaling to provide help to other aspects of the immune system. So it looks like you've got a good grasp on that as well. And then in terms of the major histocompatibility complex, so the MHC complex is what makes our immune system unique to each individual. Do you agree with this? Yes, it is. It's what gives us that unique kind of fingerprint, as it were. And then the T cell receptor only recognizes antigen, not the MHC. That is, of course, false because it's recognizing the MHC as well. And then do you agree with the following statement? Superantigens are recognized by T cells after first being processed. So they're not processed. So that's what we were trying to talk about, is that it doesn't go through that antigen presentation thing. It's kind of bypassing that whole antigen presentation. So that is that. In terms of reading and stuff, there's some resources on Canvas. There's also a little kind of activity, like a pair share activity, if you want to work in a group of people and just have a go at some of those questions. Likewise, that slide at the beginning, you can have a go at that. And then I think there's an announcement as well.
Hi everybody, my name is Sarah. I'm a third-year biomed student and I'm the event representative for the Bain Life Sciences Society. Next week, Wednesday, the 21st, from 3 to 4 p.m. we're going to hold a LinkedIn workshop on how to build a LinkedIn profile. So this is for Bain Sci students. You're all more than welcome to come. We'll be posting about where the workshop is going to take place on Monday. But yeah, just wanted to let you know, tell your friends, bring as many people as possible, and it's a really, really, really good opportunity, especially as second-year students and as Bain students, to get your LinkedIn profile all sorted out, because you can get internships through it, placement years, and even like postgraduate opportunities. A girl from our society, from Masters, she's already graduated. She actually got her master's through her LinkedIn profile. So yeah, it's just a good way of putting yourself out there, especially as a second-year student. So it's next Wednesday, 3 to 4 p.m. And our Instagram is Sussex_Bainside. If you want to go follow us for more updates and where the event's going to take place. But yeah, I look forward to seeing you all there. And good luck with this module. It is. But he's great.
All right, thanks.
No worries.
All right, thanks a lot, guys. I'll see you on Monday then next week.